Hello and welcome back to Teach Me in 10. In this episode, we're joined by Dr. Hagen Gegner, a scientific communication and project specialist at CTC Analytics AG. We'll explore microsolid phase extraction, or µSPE, an automated miniaturized sample cleanup technique that's revolutionizing analytical workflows. You'll discover how this innovative approach aligns with green analytical chemistry principles while improving efficiency and reproducibility across food safety, environmental, and clinical workflows.
Thank you so much for the lovely intro. Today we're diving into microsolid phase extraction, or µSPE. We will explore why this automated micro approach is so critical and see how it's being applied to a variety of analytical challenges. To set the stage first, let's talk about a major challenge in our field, reproducibility. A recent meta-analysis of clinical studies published in Trends in Analytical Chemistry found that a staggering 85% of proposed biomarkers are just statistical noise. Why is this happening? Well, a recent review of 80,000 metabolomic studies, detailed in analytical chemistry, points to a key issue: small sample sizes. As you see in the figure, from a subset of these 80,000 that mention sample size, over 70% of studies used a sample size below 50. When we now combine these small underpowered studies with the high variability from manual step-by-step lab work, we get data that's hard to trust and often impossible to reproduce. How do we fix this? To come up with robust and reliable biomarkers, or more broadly speaking, data, we must first conquer this type of noise. This requires two key things. First, well-designed, adequately powered studies, and second, just as importantly, rigorous standardization of the entire analytical workflow. We must find a way to minimize the technical variability introduced during sample preparation. This is where automation becomes essential. This image shows many aspects that you are very familiar with in the lab. You have your sample transfers, your pooled QCs, your dilution series, adding internal standards, but also finally the extraction or sample prep in itself. We are now ignoring the sampling. This is also, of course, a source of variability. Every single one of these steps is a source of potential human error, a slight variation in pipetting, timing, or mixing, some sort of misplacing, or forgetting to add something, or adding it too much. I think these are all aspects that we know. Here, platforms like the PAL system can help with every aspect that we just mentioned, while also taking care of the online injection into your LC or GC. This is not just about higher throughput, so measuring day and night, but it's about the improvement of your data quality and the overall reproducibility, as you're not dependent on the person, on the technical expertise of that one person. This brings us to automated µSPE. This is our solution for a greener, smarter, and more reproducible cleanup. On the right, you see the PAL system's µSPE tool in action. And on the left, you see the PAL µSPE cartridge itself. You see a cross-section of that. You can see it features two filter frets that sandwich a sorbent bed. This sorbent can be adjusted depending on your workflow, on the chemistry that you need. Furthermore, the cartridge is designed to penetrate pre-slit septa, but it can also deliver the cleaned extract directly into your LC injection ports. That's pretty cool. So you can exclude one additional step there. So this is what the automated µSPE workflow looks like on a PAL as a module. You start here with your raw extracts. The PAL automates the next steps, taking the raw extract and processing it through the conditioned µSPE cartridges here. This is the core cleanup step, right? Then the PAL can pick up the cartridge and move it to the elution tray right here. Here it collects the final extract, which is then ready for GCMS or LCMS analysis or additional sample prep steps. Before we look now at the applications, let's look at the solvent inside the cartridge, because depending on what you put inside the cartridge, can be used in different modes. First, there is the pass-through mode. Here we use a solvent to trap the matrix interferences, allowing your target analytes to pass right through for collection and analysis. There would be this side here. On the other side, you have the bind and elute mode. This is the opposite. We use a specific solvent, like, for example, weak anion exchange or WEX sorbent, to capture and concentrate your target analytes, like the PFAS that are shown here, while the matrix interferences are washed away. We then elute our clean and concentrated analytes afterwards. Here's our first application showing the pass-through mode in action. Hovering at the top here, slightly translucent, is a traditional catcher's workflow for pesticides in orange juice, followed by a dispersive SPE cleanup step. In the more bright colors here, you see the catcher's workflow turned into a fully automated µSPE workflow based on the work by Hans-Joachim Hübschmann. We see a nice reduction in solvent and sample consumption, going from several milliliters of extracts down to just 400 to 500 microliters. The automated platform is also versatile, able to handle both liquid samples, like this juice, but also solid sample extracts like shrimp. We'll see that in a second. But most importantly, for any routine lab, cleaner extracts mean less instrument maintenance and more uptime. If you're interested in this, I referenced all the applications at the bottom each time, so you can look them up and see them in more detail. Now for a major global challenge, analyzing PFAS, this time in shrimp, as I mentioned before. This application node is a collaboration with Agilent, showcasing a fully automated workflow that meets very strict regulatory requirements. The PAL system automates again the entire process, from the initial catchers extraction, vortexing, centrifugation, to µSPE, and finally to the addition of internal standards, and then also the injection into the LCMS this time. Beforehand, with the orange juice, it was GCMS. This solution provides high sensitivity and reproducibility, and I think this is exactly what you need for these types of persistent environmental contaminants. As a side note, which is quite important, and I have it here in our little cartoonish bubble that pops up, we do have PFAS consumables and analysis kits to reduce the background of PFAS in your analysis. These kits focus on specific tubings, vials, syringes as well. These changes are very important as they overall reduce your background and make your overall analysis more robust as well. Building on that, and now we're getting really complex, we have the fully automated EPA 1633 workflow. This is the US EPA's method for analyzing 40 different PFAS again, in wastewater, soil, tissue, like fish or shellfish. This workflow nicely demonstrates the bind and dilute strategy we saw earlier. Here again, it's the WEX cartridge, so the weak anion exchange cartridge or sorbent. It captures and concentrates the PFAS, effectively separating them from the sample matrix before analysis. You can also see how complex it can get with several steps being done also simultaneously. This is done through overlapping tasks. And it increases not only efficiency, but it just makes sense, right? If something is sitting in the centrifuge, the part doesn't have to wait for 2, 3, 5 minutes of centrifugation. It can do other things. So these tasks can be overlapped, and you can streamline and make your automated method even more efficient. So to summarize now, automated µSPE is a very powerful and flexible tool for your analytical toolbox. These µSPE cartridges adhere to green analytical chemistry principles by minimizing solvent consumption and waste. Their design allows for easy automation and incredible versatility. Whether you're doing a pass-through cleanup for pesticides, as we saw in the beginning, or a bind and dilute workflow for EPA 1633, or even, and I haven't shown that, complex lipid fractionations and metabolomics cleanups. If you're interested in that, follow up on our website. We have some nice posters on that, where our collaborators separate lipid classes with these µSPE cartridges. By standardizing our workflows with robust automation, we can finally move beyond the noise and start generating reliable and reproducible data, while also ensuring traceability. On top of that, it also makes the translation and adoption of these workflows for other labs or onto other setups in your own lab way more easier, as it's just a script and the modularity is quite easy to establish then. Well, that brings me already to the end of my µSPE talk. Evidently, I could go on when it comes to the applications, but I will stop now. If you're curious about some of the details, please reach out to us, but also check out our resources. On our webpage, we have a content hub that is full of app notes, posters, and also other webinars. Well, with that, thank you for your time and attention, and thank you for having me today.
Thanks for joining us for another Teach Me in 10, brought to you by Pal System, providing modular automation platforms that streamline laboratory workflows and empower scientists to tackle complex analytical challenges. To learn more about automated µSPE or the resources discussed in this episode, check out the links in the description. And don't forget to like and subscribe to stay up to date with our latest releases.